I'm really excited to introduce Dr. Richard Gibbs, who's at Baylor. Um, as as you know, he's had a really amazing career and a huge impact in genetics in a lot of different fields and a lot of different projects. He originally got his PhD from University of Melbourne, and first came to Baylor as a postdoc, and later founded in 1997 the Human Genome Sequencing Center at Baylor, which was one of the five main groups that completed the Human Genome Project. He's led and contributed to a huge number of genome projects like HapMap, TCGA, 1,000 Genomes, and many non-human genome projects. I saw Drosophila, mouse, and even a flower beetle in there looking through his publications. So there's a huge number of clinical discoveries. He's been involved in a lot of genetic diagnostic methods, as well as basic science. So there's really too many accomplishments and appointments for me to list, or we won't ever get to hear him talk. So I'm really honored to introduce you and excited to hear your talk and look forward to hearing what you have to say. And so I will then pass it on over to you, Dr. Gibbs. Thank you.
Thanks, Michael. Let me share my screen. And let me know when we're good to go.
Looks good to me.
Okay, thank you very much for the invitation and for being here today. I have no disclosures, although the way things are going, I'm actively trying to develop some. Move to the next slide. So today I want to talk about a little about the status of common complex disease genetic studies, and then where we're at with rare Mendelian disease studies. But really, my aim is to lead you to thinking a little about somatic mosaicism, which many of us are working on now as a potential driver of important issues in genetics. So to start sort of the big picture part of my presentation, I want to explain how it is that people from my vintage think about things in terms of genetic discovery. Really to set the stage. Those of you who remember, in the 80s and 90s, the way that genes were discovered in association with disease was primarily through linkage, followed up by genomic mining without a complete genome sequence to work with, in order to discover alleles that actually had functional significance. And it was done by finding the allele that segregated faithfully in the family and was represented by the linkage to be in the right place and had a function that would most likely relate to what was being needed. But the point I want to make is that in that period, the sort of Mendelian genetics practice was this process of looking for genes, assembling families, establishing linkage. And once that was done, there'd be a huge race between competing labs once the knowledge of where in the genome to really look. And then once the allele was discovered, there was a declaration of, aha, we have it. And that process usually happened pretty quickly after the linkage was established. Once the gene was found, then this opened up the whole arena for studying the functional biology of that locus. And the examples I've got on the slide here, some of you from those times will know that there are kind of interesting exceptions to this simplistic story, but they all do generally conform to this model. So that's how we grew up in the 80s and 90s. Get the next slide. But then, a little later, when the HapMap was finished and we, as a community, developed these high-throughput genotyping methods, the first GWASs came along. And we were stunned, of course, and excited to see this classic picture you see on the right from the Wellcome Trust Case Control Consortium, where those individual peaks all represent genetic signals that point to the part of the genome that must be driving these adult common complex diseases to some degree. And so we sat back at that time and thought, well, gee, if we wait this couple of weeks and a couple of months, maybe, we'll see the results and the fruits of the search in the genome for those alleles that actually cause function. And of course, here we are 2 decades later with really that story not being completed. And so this is the criticism, or the perspective, I guess I should say, that Mendelian geneticists bring to this kind of thinking. It's not a criticism of the wonderful work that's done in large-scale genetic studies. It's just a reflection of the complexity of adult common complex disease genetics, and perhaps in some quarters a failure to recognize that that means that the story's not as complete as we want it to be. So that's the perspective, and this is why when we think about what we've gotten from the Human Genome Project, where we had the initial view that we could promise all kinds of wonderful understanding from just taking this cauldron of genomic data and then letting it echo through the structure of biological science. And we could find new drugs and do personalized medicine and along the way establish the genetic architecture of disease. Of course, my point is that we haven't done that. We don't have a real handle on the clinical utility of genetic markers in common complex disease as clinical predictors. And that's the dilemma that we're faced with now. I'm not saying that the Genome Project wasn't the most wonderful thing in the world. Certainly did change everything. And down here on the bottom right, I'm showing who did the 2003 complete sequence in the different genome centers. So this has gone on since then, of course, and now looking at probably in excess of 10 million genome sequences out there, all of which have added to our knowledge of biology, and which there's some wonderful science going on, of course, but we're still stuck with this central dilemma that we really haven't found the markers that provide full clinical utility for genetics for explaining common complex disease. I've spent my time looking back on some of these data sets and scouring the literature, asking for exceptions that simply prove me wrong. And if you look at the All of Us data so far, I would say that we haven't got the yield from that yet that solves this issue. We've certainly got information about the importance of diversity, biological diversity, in giving us a full understanding of all genetic markers, both pathogenic and those ones we're not so certain about their pathogenicity. And Eric Venner and Moaz Darwood in the Genome Centre here have both looked at those two questions and published recently. And the All of Us data has recapitulated other associations that were known, but still we've got this absence of this sort of plethora of new discoveries of individual functional alleles that we'd hoped. If you look around further, and I'm a little out of date, probably 6 months out of date for looking for good stories. There's one out of the UK Biobank, this gene, this locus that's involved in type 2 diabetes, now known to be involved also in the loss of Y chromosome mosaicism, and it's a driver for that. That thing should be in the clinic somewhere. So that's a success story, but I think my general point still prevails. So we really here are facing still this giant problem of missing heritability that we all talk about, but I think don't spend enough time reflecting on. It's pretty bad. So, for example, if you have a heart attack, the family studies and the twin studies tell you that probably 30 to 50% of your risk should have been calculable. There's a genetic risk component that should be identifiable for that to happen. But the molecular data that we have in hand right now just explains a few percent of that. The gap is really quite large. So along this journey, what we've done is look for other sources of signal that might explain the architecture of common complex disease. And of course, rare variants have come on the heels of GWAS as being potential candidates. My colleagues and I kind of codified this thinking, the idea that if we looked at rare variants of high penetrance through Mendelian studies, then in certain scenarios, we might find configurations that actually really do explain risk for common complex disease. And that's represented here as the Mendelian story on the left, these compounding through burden studies and so forth into this oligogenic models, perhaps eventually giving rise to explanations for common complex disease. And Eric Bowinkle came up with this beautiful diagram here that says really those alleles can float through the population in this kind of inverted pedigree here, and through recent new mutation as well can bring out the contribution that could really be driving disease. And of course, this paper is a decade or so old. It was really only then that we discovered just how much rare variation there is in humans through thousands of genomes and other projects, and how steep this curve is. So that's been a driver of our thinking. So as a result of that, of course, we've all been enthused about the Mendelian studies we've been able to do. And some good news is that, for quite a while, they've been in pretty healthy state. And you're all familiar with whole genome sequencing and exome sequencing as modalities to boost Mendelian discovery. We did a calculation in one of these consortia. About 30 exomes get you a novel gene discovery in the Mendelian world. So you'd think with that kind of efficiency, with this kind of appetite for whole genome sequencing everywhere, that it'd be possible to fill out the Mendelian catalogue. The statistics are that, there's 20,000 genes. A little less than half of them are probably unsolvable. I'm not sure about those, but at least half are solvable. And we have about about a quarter where we've got the solution in association with the phenotype with the disease. So if we want to use rare variation and its contributions to disease as sort of the foundation of our understanding of all disease, we've really got to fill out that catalogue. Now, the bad news here, it was actually reflected by a nice talk that I heard from Jessica Chong just several months ago. She's been tracking the Mendelian discovery rates and shared this graph with me. Exome sequencing, took over, boosted the rate of discovery, but it's fallen off in the last few years. And this is actually, to a large extent, driven by funding decisions. But also some practical issues and the fact that the research cohorts of basically getting towards being exhausted, and new ways need to be discovered to do Mendelian discovery. So logically, the place to boost Mendelian discovery is in the clinic, because where, of course, you can have more facile access to individuals who may harbor a genetic disorder, and if things work out, you can also get at the complete phenotyping data through the clinical record. And if you can access that and complete this virtuous cycle that we'll talk about of the interaction between clinical and basic research activities and get through the compliance issues there, then perhaps we can use that route to boost Mendelian disease discovery. So in the light of that logic, many of us developed these internal clinical labs and methods and modalities for doing clinical data return as part of our practice of gathering research data in order to boost genetic discovery. And that's going on in many of the academic centres, including ours. There's a whole problem here that we call the catch-22 of clinical genomics. It's basically that getting these tests in the front line in the clinic at high throughput has been troublesome because the clinicians don't generally see it as the first place to go. That's even true now in most aspects of pediatrics. And of course, we need that there to get the data that we'll use to boost discovery to make it more useful so that we can have more of that activity in the clinic. That's the catch-22 of clinical genomics. So we're tackling that there in a couple of ways. And so I'll just tell you briefly about two programs. One of them is funded by the Chan Zuckerberg Initiative that I imagine you all know about in some capacity. They support education in health, community interactions in health, and also basic science. And we've received one grant from the support of this for a program we call the Texas Medical Center CZI program. And so the basic idea here is that we would boost Mendelian discovery in the clinic by having detailed and a deep and longitudinal interaction with families who present for pretty much any clinical condition for which there can be any suspicion of a genetic contribution. So our colleague Catherine King leads that at the Texas Children's Hospital, and she's recruited about 100 families so far in the year or so this has been going towards our first objective milestone, which will be 200 families, and has these families in tow. We're diagnosing, the usual 30 to 50% of those and finding known loci and alleles that explain their genetic condition and returning those data to the families. But more significantly here, we're able to take those data and samples and cycle them through our local functional biology colleagues, primarily with Hugo Bellin, who mainly through work in the UDN Consortium has been able to develop and demonstrate a Drosophila system for analysing any human variant that occurs in an ortholog that is present in Drosophila. So, the majority of genes we find are represented in that way, and he has this this system that I won't try and explain to you today, but basically swapping out the human gene for the Drosophila gene and looking for complementarity through a variety of phenotyping steps. That's working pretty well. Here's just one illustrative example where there's a known condition called Mitchell syndrome, driven by gain of function in the ACOX1 gene, and he's also studied deficiency of that gene in the fly system. And through the extensive phenotyping, looking at the common intermediate phenotypes, oxidative stress, he's at more elevated oxidative species in different cell types, he's identified the link between this lesion and neurodegeneration in the flies. So we're very bullish about this because it's a spearhead really linking rare and common disease with childhood pediatric disease with adult onset disease, and it's mediated by these lab studies as well as by the connection to the families and longitudinal studies. Now, the second attempt to do this is really part of our overall effort to kind of grease the wheels of getting genomics into the clinic. And this is a published study, so I'll just give you the highlights here. We began this just before COVID. Was the idea that if we could introduce technologies that would give genetic data that explained cardiovascular risk in our local ambulatory cardiovascular related clinics, and we could get useful genetic information, then we might encourage all of our physician colleagues to ask for these kind of tests. Meanwhile, we could use the data for research. So we established a system where we had the research coordinators in the clinic talking to the patients, getting the blood, getting the sequence, sending it back to the patients. And here's the report that we gave. There was a lot of work building all these tools that are necessary for this kind of thing, but are kind of blue collar, so they're not so exciting to talk about. But mechanically, a lot of work to get an ordered, compliant report back to a patient cohort so that you could justify doing the genetic testing. And basically what we decided to tell them was any information about highly penetrant Mendelian alleles, a little bit about polygenic risk, and I'm happy to talk about polygenic risk, but we're not huge fans of that here, and some other genetic data that might be useful for them. The stunning finding from this study was that nearly 10% of the people who were presenting at our local ambulatory clinics actually had Mendelian conditions that had prevailed through their life, not previously diagnosed. This is a very high number. It's 1% in the general population. And so we'd managed to enrich by simply going to people who already had a clinical finding of some sort, not a severe one, these aren't people in the hospital, but one which brought them to the clinics. So that was quite stunning and told us that we might find other genetic signals in those individuals that are not diagnosed immediately. Overall, we like to cite that 30% of the individuals had genetic information that impacted their care in this pilot. But to me, the real stunning thing was this 10% that have the severe Mendelian signals. So we followed it up with other pilots, which I won't give you much details of, but I'll have to tell you that they're not all giving this high 10% hit rate, unfortunately. Down in the Rio Grande Valley, we have hit rate of 3 to 5% in a screen that's the same cardiovascular risk genes plus some diabetes genes. It's a potentially different clinical scenario, but still 3 to 5% is an enrichment over background. And we're expanding this right now within the local care group to a larger extent and discussion really about the resource base versus how low of a hit rate we can tolerate while still declaring this satisfactory. But the point I want to make is that as we're trying to expand this catalogue of Mendelian disorders, we are recognizing that these adult ambulatory clinics do remain a major opportunity for Mendelian discovery. So that's where we're headed. But now none of this actually explains this basic problem that I started started to discuss today, which is where is all of the rest of this genetic signal? We're missing it. In all of those Mendelian studies, and even the ones where we've been able to follow up the single gene discovery in cohorts where we can establish burden studies, we've certainly not solved the grand question of where is most of the missing signal. We've found interesting results from burden studies where we've got contributions from multiple carrier alleles in the same individual giving rise to a phenotype, but this isn't it. We need more. So what are the other explanations for where there might be a missing heritability? Well, I think some of the people on the call here today are more than familiar with looking at structural variants and other kinds of genomic signals that might be responsible for the missing genetic signal. Many interesting findings, not enough to really explain what's missing. Other explanations, of course, are that we're misguided in thinking that there's so much genetic signal, and in some instances, just that there's such extreme polygenicity that we won't be able to easily find it by molecular methods. And of course, a lot of work going on gene regulation, trying to find the missing signal. But what I want to move to now is a discussion of potentially that somatic mutation is a factor we haven't really thought enough about, and for which there's a great opportunity to discover its contribution. Now, it seems illogical to talk about somatic mutation as potential for the explaining the missing genetic signal in common disease, because by its very nature, somatic mutation occurs after you inherit your parent's genome and wouldn't figure in population studies. So it requires some predisposition to these genetic factors, and it can't be as simple as something like a mutator phenotype and a mutator driver, because that would have been found already probably in other accompanying studies. It's got to be something much more subtle that we need to eke out. Now, what do we know about somatic mutation? Well, we know that there's a lot of them, that as you develop, from your gametes through your single cell stage into your adulthood, the mutations occur at somewhere from 1 to 10 per mitotic divisions. You get some de novo mutations you inherit, maybe 100 per individual, but after that, 1 per 10 per division. So depending on which cells you look at, which cell lineage, how long that lineage is, and its replicative state, you can get from a few to an awful lot of somatic mutations per cell. We also know that somatic mutations certainly cause non-malignant disease. And of course, in the discussion today, somatic mutation is synonymous with somatic mutation and benign disease. We're not talking about malignancy here. So, there was a splash 6 or so years ago, I think, with the discovery of the Proteus syndrome's association with AKT1 somatic mutations, and also Vexus, an immune disease that was found in a number of individuals. These are somatic disorders where you don't have a germline equivalent that overlaps with the clinical syndrome. But there are many others where the somatic event is a phenocopy of the inherited form and can contribute to phenotypic variability. And in fact, if you go to OMIM right now, there's 250 annotations in OMIM, but I would say that OMIM hasn't pitched itself directly at this question of how do we catalogue somatic disease, and we still have some way to go there. Now, summarising all this would say that the knowledge state is that somatic mutations definitely can cause non-malignant disease, but the extent to which they contribute to that is really overall not known. So what we need to do first of all is really identify the full range of contributors to somatic disease through development of new methods, and that'll allow us to characterise some of the mechanisms and hopefully lead us back to this question as to whether there's a predisposing factor or some heritable conditions that can contribute to the modality of development of somatic mutation-driven disease. Now, it's a pretty hard problem. And why do I say that? Well, if you think about this curve here, the number of occurrences and the allelic frequency, VAF here means it's basically how many sequence reads you get that represent the mutant allele. So an embryonic mutation would have a high frequency because it's happened early, and any tissue you sample later, you would have a lot of representation of that somatic variant. Alternatively, signatures, where, I think you've probably all read a lot about, if you take lung samples from smokers, you can see the signature of smoker-driven mutagenesis. If you take skin cells, you can see UV-driven mutagenesis. This is actually a relatively technically easy signal to discern. You can do some sequencing. It doesn't matter how much coverage you get of the genome. You sample the whole tissue sample, the whole homogeneous sample, and you can easily derive a signature. What's a really tough problem is to have a method that is accurate enough and can be tractable enough to sample deeply into local tissues, local clonal events, and extract these mutation signals that aren't so ubiquitous, but they're everywhere, but for which you've got to do deep sampling. So it's a tough problem. So now I'm going to tell you about 4 vignettes from our work that sort of infer on this and bring us back to the technical challenges and where we're going with those. Now, the first one is back on that heart care study, the one I mentioned just a moment ago, where we got 10 to 30% of people with a genetic signal, where we asked, what about the other people? Well, because we'd done this study with a panel, we had deep around 300x sequencing on all of the sites that we'd sampled in that 158 genes that we were examining. So Verena Chander, a graduate student then, she set about burrowing into that data and asking were there any mutations that were at a lower allele frequency that might represent somatic mutations that were present in those individuals in the blood samples. And so she developed some statistical methods to look at this lower allele fraction. Here's the heterozygotes, here's the homozygotes, and this is blown up on the right here. And she burrowed into that population to look for potentially impactful variants. Of course, she found a lot in the first pass, thousands, and set about filtering those down based upon some of the usual things you would apply to analyze potential pathogenicity of any allele: population frequency, the quality of the initial signal, and the prediction functional effect. And you can see here that she still found many potentially functional variants amongst those. And in fact, when she drilled this down across the genes that were analyzed and asked, did she find potentially impactful somatic mutations in those, there were 9 gene candidates that arose. Some of those were in genes that are associated with arrhythmias, some with structural malformations in cardiovascular structural patients, and others in amino acid metabolism. Now, this is the list of genes and the list of pathogenic or likely pathogenic markers that are found there. This is not simple CHIP. There's a more complicated discussion to have about that. CHIP, clonal hematopoiesis of indeterminate potential. These, we believe, are true somatic variants that reflect potential mutations that are linked to the disease. Now, there's a lot of follow-up and other questions this begs, of course, including that this is a blood sample, how does this relate to the organ burden, etc., and that's our follow-up. But the takeaway from this vignette for us is that the unsolved adult cardiovascular risk patients do bear a significant number of putatively detrimental relevant somatic variants. These then can be considered, I think, as part of a clinical follow-up and a screening program. Now, the second vignette comes from Eric Venner, who, based on the earlier heart care work, started to think about looking for signals that might be present in some of the large sequencing cohorts that we all have access to. Eric has been studying epilepsy in a more constrained population, looking for familial forms and what signals they can find in the syndromic and also in the idiopathic cases that present with the collaborators we have at the University of Texas. Now, he asked that question, what about these same genes? How are they, what's happening in large cohorts? Can you find a somatic signal? So he looked in the All of Us data set in this 250,000 whole genome sequencing freeze. And now remember, this is whole genome sequencing now, not panels, so the coverage is a little low, it's like 30x. And he burrowed into that to see what he could find. Now, the short conclusion from his study, oh, I should tell you what he did. He looked at 110 genes initially, added a few more took some out, ended up with around 100 genes, and looked at potential regions in those loci that could represent pathogenic sites. The short answer is, when you compared the entire All of Us data set with those who had seizures in their metadata, indications that they might be part of an epilepsy cohort, he could not find a significant difference in the number of loci in the epilepsy gene set which had a potential somatic burden. However, he did find some nuggets, that is, individuals where there was clearly a signal that had been missed. For example, this middle-aged man here who had a long history of seizures and who has a fairly significant representation in blood of an allele and a locus which is known in the heritable form to definitely cause a severe neuronal disease with seizures as part of the phenotype. And there's one or two other stories to add to that that I don't have in the slides that he has since identified. So the conclusions from vignette number 2, looking for somatic signals, are that there's individuals with low-level seizure variants that do suggest mosaicism, and that if deeper sequencing was done, we might find some more, and that might then represent just another screening opportunity. Okay, what about vignette number 3, looking now for more signals related to somatic variation leading to disease. In our Grigor cohort, which is the current iteration of the National Human Genome Research Institute's Mendelian Discovery Program, for which some people on the call here, I hope, are members of that program. I know some are. Locally, Jenny Posey is our, and Jim Lupsky are my colleagues working on that. So we have a cohort where about half of the samples don't have a diagnosis or a candidate. I'm sorry, half of them have a diagnosis, a quarter don't have any clue of what we've got, but another quarter do actually have a candidate that might be informative. So these are the individuals that have been examined to ask if there might be some mosaic variation in there. So Annie Rivera, graduate student with Jennifer, and Chris Kakowski, a faculty member in the Genome Centre, have done a sort of large pilot here, looking across probands, 120, in those unsolved individuals. And this time they haven't used the sort of homemade statistics that Varuna did. They went to the high sensitivity dragon calls, which reveal these low allelic fractions. And then they've filtered down from them to see what they can find. They found a number of probands with significant allelic fraction that look like it should be examined, and mining into those, they've solved 3 new cases at the time of this slide, a couple more since. So these cases, you can see individual genes represented here, and you can see sequence read panels here with a few of the reads representing the mutation. And these have been validated using ddPCR, following up, looking at the low signal in the proband. So the conclusion here is that incremental solving of Mendelian cases can also eke out some of these mosaics and include improve the inventory here. Okay, so the 4th vignette here, I want to talk a little bit about Shire-Gibbs syndrome. So this is a disorder we've taken a deeper dive into. It's a disorder that we first discovered in my grandniece when we first developed the clinically applicable exome capture methods and started applying them to new mutation discovery. I got a call from my sister saying her daughter, my grandniece, Molly here, had a developmental problem. We sequenced the family, found a de novo mutation in a gene that we didn't know much about, and then found 3 others in our growing clinical database at that time, and defined a new disorder. So the gene is AHDC1, HE hook DNA motif binding containing protein number 1, and it's a single large coding exon. And all of the initial cases were, truncations. They were de novo dominant mutations, typical of a severe neurodevelopmental disorder with these generalized symptoms. Now, we've gone on since then to build a registry, and we have over 500 families worldwide now that we know about, and a little under half of them are actually in our registry with full clinical data. And so we've been able to do some lab work and also some mining of the clinical spectrum in order to draw some conclusions about the underlying biology of the gene. Now, for the most part, the individuals have these protein truncations. They have frame shifts or stop-gain mutations that give this dominant effect. When we delved into the relationship between the length of the residual protein, that would be depending on the position of the truncation, we're able to establish some phenotypic correlates with the length of the protein, both at the level of the fully developed individual, but also in transient expression experiments where we saw different cellular effects that are associated with the different lengths. Now, I'm going to not dwell on that too much. I will mention that there are non-truncating mutations and a minority in these individuals. And like every Mendelian disease, when you delve deeply into it, you find exciting things. Here we find some missense mutations that are de novo, that are pathogenic, others that are not, depending on the functional sensitivity of along the protein. And Varuna again studied one individual with a smallest deletion that we knew at the time, and found that the phenotype was a little unusual, was a little mild, and the deletion took in adjacent genes. Turned out AHDC1 had some compensating expression in that individual. And so the blood level of AHDC1 was almost normal, while the adjacent genes involved in the deletion were appropriately damped down to 50% of their normal expression levels. So there's some kind of genetic compensation which tells us about the potential regulatory aspect of that locus. But I digress from the somatic mutation story, because what I want to tell you is this. In a typical individual with Gibbs syndrome, what happens is they present with these generalized symptoms of developmental delay and hypertonia, and then somebody does a whole genome sequencing or an exome sequence or a panel, and in the AHDC1 gene, they see 50% of the reads are wild type or normal, and the others have a mutation that reflects a truncation of the protein or allows you to predict the truncation of the protein. And this is then a confirmed molecular diagnosis of Gibbs. It's the majority of the cases. So we say that the mutations that do lead to truncation of the protein are really considered pathogenomic for the disease. You've got these mutations, you've got the disease. You've got the disease, most likely you have that mutation. So imagine our surprise when we learned of this individual who had an unusual molecular diagnosis. This individual had the typical clinical features, developmental delay, hypertonia, etc., and only one disease gene candidate, AHDC1, but the alleles were mosaic. Now, what do I mean by mosaic? There are actually three types of sequence reads. There was the normal sequence reads through the locus, about half of the reads were that way. But then the remainder of the reads were more or less equally divided between two different frame shifts. These frame shifts were in these similar sequences here. They were quite close together. The nomenclature suggested they were 3 bases apart, but actually we deduced later they're probably 1 base apart. So this is called double mosaicism, and some of you may be familiar with it, but it's actually not that frequently described a phenomenon. Now, we followed this family up with other sampling of other tissues, with maternal sampling, and with long reads to complement the short reads, and we're able through that to phase the variants and to establish the paternal origin of the variation, and to convince ourselves that the two variants were always found on different sequence reads. Now, I'll explain what that means in a moment. It was interesting exercise to establish the paternal origin because we didn't actually have the father in the family, but the mother had a common haplotype, and the mutations both occurred on a rare haplotype. And so we deduced that therefore that must be from the father. Here's what is going on in this individual. So the damage comes in somewhere between in the paternal lineage to the genome. And then in the zygotic stage, there's a potential repair event, but the repair event doesn't resolve completely. It resolves differentially with the lesion and differentially into the two daughter cell types. So half of the cell types bear this frameshift type number 1, and the second frameshift type number 2. And through more deep sampling, we established that these occur pretty close to equally in this individual. Here's a sort of DNA version of the same story. Here's the maternal duplex, and here's the paternal duplex, the damage occurring here somehow. And then that resolves into these 2 cell types. You see we've got TAGCC, and here we've got TAGGC. So the mutation is likely in adjacent bases and is likely due to the way that this event was resolved in DNA repair. So it's interesting to speculate what kind of repair and what kind of damage this might bring about. Of course, we can't really know, and it's not a CPG, but there may be some local deamination, some deamination of the C. There's potentially a hairpin structure here that might aggravate the repair to produce this unequal, this inappropriate resolution. And there's a lot of discussion now about this ribonucleotide insertion repair driven by nucleotide pool perturbation, but we can't really know in this individual. Okay, so what's so interesting about this case and about somatic variation? Well, the significance here really comes from the fact that in this case, there's about a 50-50 representation of the 2 cell types in mature blood. Now, those of you studying hematopoiesis know that the events prior to the formation of the hematopoietic stem cells involve some kind of black boxes. These cells arise in the endothelial cells in the lumen of the blood and in the bone marrow, but how you get there from your zygotic state is really something of a question. And interesting paper we read and discussed with Alexei Abyov recently about how individual blastomeres can be segregated out and to form these different somatic lineages. So the blastomere is the very early cellular formation post-zygotically before the blastulation, and these individual cell groups can have individual fates. His thesis is about, and his use of early mutation mapping is about how there is an equal contribution of different blastomeres to germline, which is a potential evolutionary mechanism to preserve the integrity of the germline. But our finding really is that if you think about the 50-50, it's prevailed all the way through to mature hematopoietic stem cells, and that means something about the bottlenecks in these lineages. Now, that makes you ask the question, what about the rest of the double mosaics? Because there are some that are reported. Here there's the cases that I was able to find in the literature, and in these different disorders, there's been different measurements of the representation of the 2 types in different tissues. And it's a bit hard to read this table, so I made these little charts. And so this is our case today with Gibbs, where there's about 50% of the wild type and the other 2 types are represented more or less equally. And you see that here in this neuropathy and in more or less in this Marfan's case. But in these other cases, you see a skewing of one versus the other. And delving into each of these is informative. Sometimes the reason for the skewing is not related to simple selection of what somatic lineage is represented, because there is a much more complicated genetic story in the family, like in this Cornelia de Lange case. But the point of this is it illustrates the power of these markers to allow this tracking through somatic variation. So it would be great if these double mosaics were common, because then, the ones we've observed and ones that get reported in the literature are in the context of a phenotype. But what if we were not scoring enough of them because we were ignoring the ones that weren't related to a phenotype? Aniko Sabo in the Genome Center went then and looked at a number of the trios we have in our collection and asked the question of if she looked at de novo variant sites that involved multiple events, potentially multiple events, did they occur on the same DNA sequencing strand, or did they occur on different sequencing strands? If they're on the same DNA sequencing strand, it's just some complicated event that's happening in normal DNA repair, and the things aren't segregating the way they are in double mosaics. But if it was on different strands, then we would have more double mosaics to do this kind of early lineage mapping. Unfortunately, they're all on the same strand in these collections, suggesting that these are relatively uncommon events. So to summarize all this, what I've been telling you so far and through these 4 vignettes, the common and rare disease mining shows examples for non-malignant disease-associated somatic variants. So it's a fertile place to go to look for more events. If we find them, we can apply them to deeper diagnostic screening, and we can use them as somatic variants to map cell lineage in different scenarios. So it'd be great if we could do more of this. So we can ask ourselves, what's missing here? What would be the ideal assays for doing more somatic studies? Well, you can imagine, first of all, we need methods that can get into this part of the frequency curve that's the troubling part that I talked about at the beginning of this whole discussion. Things that went beyond signatures or simply very prolific mutations, but got into rare events with high precision. We need access to fractionated tissues to be able to do that from both normal and disease states. We need intimate knowledge of the cell lineages so that we could map the molecular anatomy of organs and cell lineages back to what we find in these somatic mutation detection tests. And we should use gene expression patterns as part of that lineage mapping. Of course, we need those technologies such as deep sequencing and duplex sequencing and a lot of single cell sequencing to do this. And we needed methods to develop that. So happily, in anticipation of this need, there's a Common Fund program that was launched a couple of years ago. It's called SMAT, the Somatic Mosaicism Across Human Tissues, and its aim is precisely to fill the gaps in all of the slide that I just showed you. It's to understand somatic variation, discover mutations, develop tools and technology and infrastructure to do it. So it's the somewhat familiar distributed consortia, and we have genome centers, data centers getting the material from the tissues at 5 sites, including in Seattle and here in Houston, and also in the Northeast. There's a tissue procurement center, and there's a data analysis center, and there are multiple groups that are tasked with developing the new technologies and feeding them back into the enterprise. Broadly, the goals of the consortium are to build a reference catalogue of the variation in 150 donors. Of course, post-mortem. These will be 10 to 19 tissues per donor, using all of the sequencing methods and all of the new technologies to get the data. The donors themselves are what we would might call the healthy deceased, in other words, excluding cancer and infection and other chimeras, from diverse ancestries, equal gender bias, and adults, all collected within 24 hours of circulatory arrest. And these are the multiple tissues representing all germ types, and also some clinically identifiable, accessible tissues, so we can correlate broader studies later back with what we find in all of these. So as I said, the goals are to improve the technology and solve the issue of how do you find these rare variants and decide if something like this here, this C, that's next to that's a rare variant, an allelically rare variant, if it's real or not. And how do we do that through all of these methods, and how do we build a standard catalogue around that? And if you go to their website, you can see that, that there's 2 websites, but the DAC is doing a great job describing the progress. The initial phase, we work with cell models as a group to standardize the methods and tissue homogenates from the very earliest donor tissues. We're just now moving into these donor tissue sets to establish the catalogue. And what this nice tool does, it's live, I'm showing you a static version, is you can go to each of the contributing centers and click and find out which sequencing technologies and which assays they're using, which data type they'll be generating. And so we're really beginning to crank this stuff out now and to look at it. So to give you an idea of the flavor of the methodology, I'll tell you just 2 things. One is developed by our own collaborator here at Baylor, Chuck Zong, with his student analysis done primarily by his student Yang, which is a new version of duplex sequencing. Duplex sequencing, developed at the Sanger Center, is a methodology where essentially you have knowledge from barcoding of the Crick and the Watson strands. And if they don't match, you're looking at an error. If they do match, you're looking at a true event. that was present in the original molecules and not something that was produced by subsequent amplification of the molecules. And refinements of this in different quarters include Chuck's work, which is using transposons to get a more even genome coverage, and additional oligonucleotide molecular trickery to enhance in 2 stages to enhance the ultimate accuracy of the region that's found. And applying this to cord blood, which is the gold standard in the field for very low levels of background somatic mutation, he finds really enhanced coverage of the genome and enormous accuracy, around in excess of 10 to the minus 9 in terms of the fidelity of the final assay. And you can see his method here performing better than the alternatives. So great duplex methods now to start probing into these tissues, and that's what the group's launching into. And the last thing I'll show you is some analytical work done, because we're generating long reads from at depth from many of these samples from Fritz Sedlacek's group here. Ely has been working on adaptation of sniffles, which is a program used for assembly and analysis of long reads. He's been phasing methylation changes to go along with indels in some of these samples, and you see that in a reference sample here. But working his work with the Brain Consortium, looking for markers that relate to neurodegeneration, other neurological diseases. This is a reference sample they all work on. You can see here this mosaic detection using that method in long read data that shows a deletion largely associated with low levels of DNA methylation compared to the rest of the tissue, which suggests there's a cellular mixture, but the process has resolved the tissues. So that's my advertisement for the SMAT consortium, with much data coming to supplement our understanding of rare of somatic mutation as potential contribution to disease. So let me iterate what I've been saying for the last 50 minutes. First, the rare and Mendelian work is coming along strongly, but it was short of completing the catalogue. And this, I think, would be wonderful if we found a way to muster the community to help complete that catalogue and use that for foundationally for more rare variant studies and their contribution to complex disease. This should include then the study of somatic mosaicism as a disease mechanism. And as we do that, hopefully, as we understand more the biology of the generation and impact of somatic variants, we may discover underlying mechanisms that predispose to them, and again, link them back then to potentially heritable factors that can explain their contribution to this missing genetic signal. Okay, well, that's the end of my comments, and except for I need to thank a lot of individuals, and some of you know some of the folks here, and some of you don't. But I'll point to Donna Musney, I've worked with for years, who's a mainstay in the Genome Centre here. Jennifer, now moved to Columbia. Jim, been working with in the Mendelian world. And actually, there's too many to talk about. So why don't I stop there? And I think we've got a few minutes for questions if that works out. Thank you all.
Oh, thank you. That was really great. That was really excellent talk. So we'll have questions in the chat, and if people want to ask in person. Oh, it looks like Amy has popped up in person. Do you want to start us off with a question? Then I'll start looking at some of the ones in the chat.
That'd be great. That'd be great. I have a couple of questions, but I'll just ask the first one so that I don't monopolize it. Your study with the frequency of Mendelian explanations in the cardiac studies, those were, as you explained, you were looking for cardiac phenotype, you had cardiac phenotypes associated with cardiac genes. I was really intrigued by a paper, and there might be a couple now, looking at the frequency of pathogenic variants, known pathogenic variants in metabolic disease genes in the general psychiatric population, for example. So stuff like that really speaks to how valuable all of this could be in terms of kind of the general utility of clinical sequencing. Can you speak a little bit to kind of, what do we need to kind of make that happen more?
Yeah, I mean, I think that speaks to the power of burden studies where you have full knowledge of the range of pathogenicity of the individual contributors to the burden. And I think probably the appropriate short answer to your question is, I think there's a very high ceiling there for us to get to, lots to be discovered, but it's going to take more knowledge of individual variant spectra. We've got to do more analysis of what individual variants in genes do on their own, either through model systems or through disease discovery, before we can do the surveys in these populations and ask exactly how do they contribute to the overall burden. We're kind of working with 1.5 eyes closed in most of these studies.
Thanks.
We can go to one from the chat. So that says, how much of the Mendelian gene discovery decline is due to lack of exploration of Mendelian diseases in understudied populations, and the paucity and slower timing of experimental follow-up for candidate genes that are rare or ultra rare?
So great question. I think we can go and, when you think about the tremendous efficiency of Mendelian discovery just by sequencing, no functional work at all, because you've got a new mutation or you've just got enough individuals who segregate the variant uniquely. The efficiency of that versus the number of genomic sequences that we've contributed for these large cohorts, we've got an awful lot of discovery we can do with ever going to the experimental models. Now, I mean, the question's the right one, which is, that's fine, but if you only get one case, you can't make that statistical association. You've got to then have the functional data. So it's going to be built from both sides. But I think that we're under exploiting the power of broad, naive sequencing to drive discovery. And then we save our functional, expensive, slower lab work for the harder cases.
Claudia, do you have a question?
Well, thank you so much, Richard. It's a great talk. I love the history and the context that you put. I think I just wanted to comment, or maybe it's a question as well, like based on your talk, are you suggesting or pointing that common disease and rare disease actually help each other based on the cardiac cohort that you presented, and that we need to go to explore this aspect a little more?
Well, I think I'm becoming more extreme in my maturing years, Claudia. And I think I would have said exactly what you say some time ago. But I think to me, there's a real urgency about completing the Mendelian catalogue, and then let it reflect on what we know in common disease. Rather than, you find your GWAS signal and you scratch around looking for some Mendelian signal in that, sort of look, you hope that there's something known about a gene in there that it could be have an effect on some system that's in your phenotypic list of things that happen that you started the GWAS with. I think to me, just the right thing to do in the near term is just knuckle down and finish the Mendelian catalogue.
Yep.
And I think that'll just cast a whole lot of light. So I've tilted a little bit from I think you're more eloquent or more, from what you say.
So let me just restate that. Do you think then rare variants are the real, like, where we should go, regardless if they are Mendelian or common? Is that?
Well, yeah, but we've still got a problem. That's the thing. There are population studies of rare variants that show that this ain't going to solve this whole missing genetic signal problem. So I think we had that hope for a while, but FinGen and a couple of others tell us that that's not going to solve the story, not easily. So there's still missing contributors to the signal. Now it can be, you start adding in all of the other kinds of variants that we haven't been ascertaining properly. That can add to the signal, and then maybe this somatic story is part of it too. I think that's how we're going to leverage our way up. But it's not as simple as saying, yeah, it must be all the rare variants. But we need to understand all the rare variants and their contributions before we can really finish building out this story.
Okay, thank you.
Right. We had one other question from the chat from Ray Monnet about the mosaicism. So how important do you think somatic cellular selection of germline and somatic mutation combinations is in driving the disease? And what tissue or lineage contexts might be most revealing here?
Yeah, Ray, you nice to have your question, and you clearly have been thinking about this too. You know, we had a somatic mosaicism and disease meeting, a Keystone meeting a couple of weeks ago. It was dominated by the mTOR pathway perturbations in focal epilepsies. And some of the stories that came out of that were quite stunning. You know, tissues that outgrow but still can give rise to the epilepsy. Some discussion of metabolic sharing between cells bearing the mutation and cells that don't bear the mutation. And I mean, I think in other systems, the role of selection. I think what I'm trying to say here is that these are all important and underplumbed. We haven't got to the depths of them. And the existing stories we have, you know, there's some great stories in CHIP now about cell populations that come and go. That there's good work to be done there. Ray, I hope that's a satisfying answer. I'm just basically agreeing that you've got a great question.
Thanks so much, Rich, and thanks for coming to present this provocative talk. It was tremendous.
Thank you.
All right, maybe we can do one more. There's one more from the chat. So Claudia Gonzaga asked another question about beyond heritability. How much of the incomplete penetrance that we see do you think may be due to non-cancer double hits or germline variants in somatic mosaicism?
Yeah, so this could be the missing clue, right? You know, as we go all the way back to, you can't just say it's somatic events because it's got to have a germline handle to be part of the genetic story. And whether it's as straightforward as double hits in the sense that we think about them normally with a deletion in one haplotype and then a somatic event in the other, or whether it's some epigenetic state that occurs opposite the somatic event. That may be the story we learn eventually, but it's got to be there.
Sorry, Michael, can I pull
Go for it.
So Claudia's actually, that was exactly my second question. So I'm wondering, Richard, like how much, you know, in the case of, so for example, you know, we have an X-linked disorder in males. There's a big distribution of, you know, disease severity, and the functional assay says that this should be really low activity. And so that according to the functional assay, the really severe disease is the correct one. Should we be looking at variant frequency in those cases? Like what are people proposing in terms of kind of following up along the lines of Claudia's question?
Yeah, so I mean, is it fair to recast your question as how many of those actually have a somatic component? And, you know, if they're de novo mutations, could they be immediately post-zygotic and somehow they're being, you know, or they're being represented as pre-zygotic de novos or zygotic de novos? And so do we need to understand that tissue distribution and the variation in the somatic events before we can properly categorize the different stages of disease? Is that along the lines of what you're thinking, Amy?
I'm just interested to hear your thoughts. Yeah.
Yeah, I was having a good conversation with David Nelson. You know, there's this thing in Fragile X where you know, the sperm in some of the boys who are affected doesn't contain the expanded allele. And, you know, what's the you know, and people are pointing to selection in the male germ lineage. But it could equally be, I think, that it's, you know, it's this blastomere early, it's not, you know, every other tissue's got it, but the sperm just miss out on it. So you know, much to learn, I think, about these lineages and how they, and back to the earlier question about selection, you know, if you want to propose selection in the male germ lineage, you could just as easily propose it in the blastomerm stage. And it's certainly present in much later lineages, right? We've seen evidence for that in some of these blood studies. So once again, I'm not answering your question, but I think there's lots of stuff there, Amy, that's certainly for us to do as geneticists.
Great. Yeah, I think that's really fascinating to think that sometimes this somatic mosaicism could actually be a mutation that makes things more functional in some cases. So everything becomes more complicated.
Which is the evolving trend in genetics.
All right. Well, thank you very much, Dr. Gibbs. It's been really an amazing seminar. All right. Thank you very much.
Thanks
for your time.
all.